on these ends of the muscle and it becomes more strong. So if you have very thick bundles of those contractile proteins, you have a stronger muscle, and if you put them all in series, you get a much faster muscle. Now this doesn't just depend on this morphology, right? We also have slow myosins and fast myosins, and they differ in the rate at which they hydrolyze ATP. You can imagine if you go cycle faster through your ATP, you cycle faster through the cross-bridge cycle, and you can do more contraction per unit time. So here's the written out summary. I think that's helpful if you're studying to see if you can tell yourself this story. There's one word in there that I'm not sure that we talked about, and that's this triad here. Did we mention it yesterday? No, I didn't think we did. I put a note, but then I still forgot. So the triad is this structure here, that isn't really clearly indicated. The triad is this T tubule, and it's on either side has sarcoplasmic reticulum. So we have three things that are always hooked up together, and that's what's referred to as the triad. Right? It isn't drawn in this picture, so the picture isn't even making it very clear. So we have, this was the plasma membrane of the muscle cell, so we call that the sarcolemma. Everything sarco has to do with muscle. And it isn't just all the way straight on the outside of the cell. There's these deep holes coming in. It's not, maybe a bad word, a de-invagination. Does that make sense? I don't know who asked the question. Where are you? Yes. We call that the T tubule. I guess it kind of looks like a T if you look from the plasma membrane and then the big bottom of the T sticking in. And this goes in so quickly because the action potential, the charge difference, travels really quickly and gets down really close to where it can actually induce the release of calcium from the sarcoplasmic reticulum. And usually these things are prominent in many places of the muscle cell, and on either side you have the bags of calcium sitting there waiting to open up and flood the system with calcium. And because we then have three structures, sarcoplasmic reticulum, that T tubule, and the other sarcoplasmic reticulum, that's the triad that they're talking about. I think it might be a little here. See how it wraps itself around there? And then there's these areas where the sarcoplasmic reticulum seems to be really much more prominent and thick. In between that runs the T tubule to the inside, and then here it's a little less dense. So we really have this increased amount of sarcoplasmic reticulum within the middle, the T tubule, so the triad's coming together right there. So let's walk through the words there, make sure we understand everything that it says. The thin filament is composed of actin, triple myosin, and the troponins. So these troponins regulate the way that triple myosin wraps itself around the actin, right? And when calcium comes in, it binds the troponin C for calcium, and it causes a conformational change that moves the tropomyosin away, and now myosin can bind actin because its binding site is all of a sudden available. We get calcium into the cell because we have motor neurons, motor nerves coming in. When they depolarize, they release a neurotransmitter, acetylcholine, into the neuromuscular junctions, which open sodium channels. They depolarize a little bit, then we get the voltage-gated sodium channels that depolarize it and make it an action potential. The action potential travels down the T tubules to the triad, so where the T tubule and the sarcoplasmic reticulum meet up. Now we have a voltage-sensitive dihydropyridine receptor, which changes its conformation and pulls the ryanodine receptor open, which results in the release of calcium. Now we see when calcium binds to troponin C, we already said this, the binding site becomes available, myosin binds actin, and the power stroke cycle is initiated. Because the physical interaction of myosin on actin is what triggers that power stroke. So that's how calcium helps initiate that. This cycle continues as long as calcium is high and we have ATP. And that results in the Z lines of the sarcomeres being pulled closer together, shortening the I band. I think we went over that yesterday too. And then it describes the steps of the power stroke. And contraction will end when the electrical signal is removed, so the action potential. Calcium is pumped back into the sarcoplasmic reticulum by those SERCA proteins that are pumps. They are pumps because they use ATP to pump calcium back against its concentration gradient. Right, and so skeletal muscle doesn't contract by itself. It needs to be told by neurons to go ahead and contract, and that's how this system is regulated. That's different from smooth muscle that sits around the hollow organs of your body. That has an intrinsic ability to contract, and it can be regulated. But muscle cells, you have to tell them, you jump, and they say, how high, right? Like that, which is the machine song. All right. That was muscle. Are there questions about muscle that we should clarify before we hit endocrine? This was really a kind of a morphology combined with biochemistry lecture, I feel like. Because we really look at the structure of these proteins, so you guys are expected to know where the actins and the myosins are, and then focus on the role of calcium and ATP. That's really the main point of this set. Sure. Half an hour for the endocrine system. April will be sad. Okay, the endocrine system has some repetition from general cell signaling, which is great because repetition is the power of advertisement, so that means we will know it really, really well. The first repetition is the types of signals that we have in our body, right? We talked about endocrine signaling that uses a signal traveling through the blood to reach a target cell that's far away, and far away can be very relative. We have paracrine signaling, where neighboring cells can tell each other things by signaling. These signals are usually not very stable, or they're actively removed very shortly after they're being released, as we see in the case of neurotransmitters. And then we have autocrine signaling, where the cells tell themselves things. And earlier we also had the direct cell-cell contact type signaling that we see a lot in the immune system. So endocrine system uses this type of signaling. A cell secretes its signal into the bloodstream, it travels, and then whichever cell in our body expresses the right receptor, that one can hear this hormone signal and respond to it. Doesn't matter where you are because the blood serves everything, right? Paracrine signaling, that's really the hallmark of the nervous system, because there the nerves are touching, say, other nerves or muscle cells, really close interactions, and there's a short distance to travel for the signal. So let's summarize again here. We have two major control systems. Both of them are super important, and we describe them separately, but really they talk a lot to each other, as we'll see. So the nervous system, specialized for short, fast-acting signals, and in order to achieve this, it has many, many connections, direct connections between cells. And we talked about those because those are the synapses. The endocrine system is a slower-acting system, and it uses the blood to mediate the transport of the signals. So if some piece of my pituitary, small gland that we'll talk about, sends out a signal, it needs to travel through all those liters of blood that are in my body before it reaches something, so it's slower to get things going. The signal dilutes a lot, right? So we need to have receptors with really high affinity to make this work. But usually these are longer lasting changes that are taking place. So the focus of today's lecture is mostly hormones. The title suggests that we're comparing side by side hormones and neural systems, but mostly the focus is on the endocrine system. And we love the endocrine system because as physiologists we work with it a lot. It's important for homeostasis, to make you grow, in reproduction, metabolism, and it uses hormones. And hormones are defined as chemicals released by one cell that travel through the blood to reach a target cell far away. And far away can be relative. We'll see that there's blood vessels maybe a millimeter or two long that connect to tissues, but that still counts as hormonal signaling because it goes into the blood. And hormones are released from organs called endocrine glands. And there's also scattered single cells in your gut that will release hormones, so not necessarily a gland. And in us, many of them are organizing glands, but if you go down the evolutionary tree, you'll see that many species just have groups of cells somewhere that aren't really nicely organized as we have them. So here are some examples of how the endocrine system influences us. We can have too much or too little of a hormone called growth hormone, and so you can either stay pretty small, become a huge giant, or be fairly normal. I've never been able to figure out if this is real. This is kind of strange. Right? The proportions aren't right for what I expect in somebody with dwarfism. But anyway, so one hormone can make us grow all these different sizes. Here is a patient with a goiter, a large outgrowth of the thyroid gland. And this became much more common as we, as people, moved away from the oceans and more towards the freshwater sources that have a relative lack of iodine. And iodine lack in your diet does this to you. Your thyroid gland uses iodine to make its hormone. And if it doesn't get enough iodine, the body doesn't see enough of the signal, so it's trying to tell the thyroid gland to keep going and to grow and to make more because we don't have enough. Jet lag, there's hormones that regulate our night and day, so if you travel too fast for that system to be able to predict what's going on, what you predict is happening doesn't match up with where you are, and you have a jet lag. This one we kind of talked about too, maybe not with these different names attached to them. We mostly talked about hydrophilic, peptide-based hormones and hydrophobic, cholesterol-based hormones. And we drew a little table that said, compare these side by side, how do they travel, what's their receptor. And there will be a slide in here that summarizes that for you once again. And then there's this other group, the amines. They're kind of in between. So thyroid hormone is not as hydrophobic as steroids, but it's not as hydrophilic as peptides. So it combines some aspects of these two. And the catecholamines, there you have to think about adrenaline or noradrenaline. Excuse me. So here again, really using the two most extreme hormones in how they like to travel and how they are made. So peptide hormones versus steroid hormones. Peptide hormones are obviously a protein that gets secreted, so we all know now that it needs to go through the ER and then make its way out to the outside of the cell. Whereas steroid hormones begin as cholesterol that gets imported into the mitochondria, gets changed a little bit, it gets sent out to the cytoplasm, changed some more, and then some of them go back into the mitochondria to be finished off. So the way that they're made is very different. Because these can't really cross over lipid bilayers, we can pack them and store them. whereas the steroid hormones easily diffuse across membranes. So as soon as they're made, they are released. So the regulation is slightly different. If you're stored in a vesicle, that means you can be released through exocytosis, whereas these guys, like I said, get out of the cell through diffusion. Peptide hormones, very water soluble, so they can travel freely. These guys use a transport protein to take them to the receptor. And now some people are actually saying that those transport proteins also help with the binding to the receptor, because sometimes these guys actually appear to still have some cell surface mode of action, which is not what I'm going to ask you to remember. But there's more to it than these extreme cases of how we think stuff looks. These bind to plasma membrane receptors. I think the initial text didn't have the word plasma membrane. So these receptors are sitting on the outside of the cell, whereas the other ones are binding cytosolical nuclear receptors that will then go into the nucleus because they're trying to affect gene expression. So these hormones are slower than the nervous system, and then within the hormone group, peptide hormones are faster than the steroid hormones in how they can elicit their effect. Because generalized, these change proteins that are already there, whereas these are making new proteins. So we got some signal, we made a hormone, it's released into our blood, and then what? A lot of times this doesn't mean the end all of the hormone and its life. So it's circulating in blood. Sometimes it gets processed, and then, for example, thyroid hormone gets processed and then gets taken up in the bile and secreted into your gut, and so you poop it out. That's one way to make it less effective. Sometimes hormones are chopped up by, say, proteases that are circulating in your blood, so that's what happens a lot. The peptide hormones, they get chopped up and thereby inactivated, just to make sure that when the signal is present, it doesn't stick around for too long. Thyroid hormone has enzymes inside cells. It's secreted mostly in a not so active form. The enzymes in the cell can then make it more active so that once it reached a cell, a target cell, it really becomes active much more. And then it can find its receptor and actually do what it's supposed to do. So in us, this is where we see that the nervous system and the endocrine system actually talk to each other. In us, we have this setup with the pituitary. The pituitary makes all these hormones, and the hypothalamus is the part of your brain that integrates a lot of signals, and then based on that, tells the pituitary to go ahead and do something about it. Right? So the hypothalamus is part of your brain, the pituitary is not. It hangs kind of on the bottom of your brain, but it's outside of the blood-brain barrier. So the hypothalamus receives a number of inputs, maybe about daylight, maybe about nutritional status, maybe about it's time to go find a mate. All this stuff gets integrated, and then it tells the pituitary to secrete the appropriate hormone. The pituitary has a choice of at least 8, but many more well-characterized hormones that can then elicit the effect to change the behavior of the organism. This is just to orient ourselves. We have the hypothalamus sitting right there, and the pituitary is sitting very, very close by, but is actually outside of the brain. And the pituitary is also sometimes called the hypophysis. And if you write that down, that's, I think, helpful, because on the next slide, it uses that word again without telling you that it's actually talking about the pituitary. So the hypothalamus talks in 2 different ways to the pituitary. This is an enlargement of this area up here. We have the hypothalamus, and there's some neurons that sit there, and they extend all the way actually into the pituitary, and then they touch these little blood vessels here, and they are actually the ones making the hormone that gets released and is circulating in your blood. So it's not the pituitary that makes these, it's the hypothalamus. So these are nerve cells that are secreting hormones, so neurosecretory cells. Very direct action. The hypothalamus senses that something is needed, and actually the hypothalamus itself sends the hormone out into circulation. And that's what happens in this posterior pituitary. Posterior for the back, right? This is the back of the head here. So this is sitting in the back. There's other neurons that actually secrete their hormone here in this little set of blood vessels. Those then take it over to pituitary cells in the anterior pituitary, or the pituitary side that's at the front. And then those cells in the pituitary that have a receptor for that particular hormone, they respond by the release of their own hormone. Right? So posterior pituitary, it's actually neural hormones. Anterior pituitary is controlled by neural hormones, but they secrete their own thing. In this picture, for our inconvenience, it's organized exactly opposite, right? The posterior is now on this side in the picture. So the hormones coming into the posterior pituitary are antidiuretic hormone and oxytocin. This gives the brain very direct control over the release of these hormones. The anterior pituitary makes things like thyroid stimulating hormone, adrenocorticotropic hormone, prolactin, growth hormone, and the sex hormones, gonadotropic hormones. So the release of these guys is controlled by the hypothalamus, but they're made here in the pituitary. They're not made by these neurons. These neurons send hormones that will then travel through this little bit of blood vessel, come into the pituitary, and tell cells there to release these hormones. These hormones that are released, we call those tropic hormones, because they not only elicit some effect downstairs, but they also, whatever tissue they target, it helps that tissue to grow. And that's what we'll need to answer some of the questions that we'll get into. So this is the same kind of picture that we saw before, but now without any of the anatomy. But here we have the hypothalamus, and here listed are the abbreviations for all the hormones that it secretes. As you probably cannot figure from this, but these are mostly all peptide hormones. And the same here, peptide hormones. Right? That makes the interaction a little faster, and you can see downstream that we get those steroid hormones. But up here, where the regulation is done, they're mostly all protein hormones. So the hypothalamus will send out gonadotropin-releasing hormone. There are cells in the pituitary that have the receptor for this particular hormone, and those cells make FSH and LH, and then when they see that signal, they go ahead and secrete that into circulation. Growth hormone has this dual regulation where growth hormone releasing hormone will tell it go ahead and release, and somatostatin will tell it go ahead and don't release, stop it. Thyroid releasing hormone will induce the release of thyroid stimulating hormone. Dopamine actually continuously inhibits the release of prolactin. Right, so there's often a stimulatory effect, but we have these two examples here where the hormone from the hypothalamus inhibits the hormone from the pituitary. All right, finally, clicker question.
It looks like it would be because the GH is growth hormone, and RH is.
I forget. I said like the beginning of the quarter.
You said that?
Sorry about that. They keep calling me with some Chinese advertisement, and I can't tell them to stop. All right, I think we're about there for the number of people that are going to vote.
It's probably. I think we just saw it right now.
Yeah, it's fine. We'll change it. Change it.
I just said good.
Alright, so we want to increase the release of growth hormone.
That's close.
It makes no sense to increase the release of gonadotropin releasing
close.
hormone because that's not going to find the right cells. To increase the release of growth hormone, releasing hormone would definitely help. Somatostatin acts on growth hormone, but it's going to actually inhibit its release. So using option C is not going to get you more growth hormone. And dopamine actually acts on cells that make prolactin. And if you increase it, you will actually decrease the release of prolactin. So answer B was the correct one. We're going to skip over this slide in the interest of time. So you don't have to worry about it. But basically, first order loops make use of only one hormone. Second order loops, there's 2 hormones. Third order loops, there's 3 hormones. And then direct loops don't even worry about the brain. So for example, the pancreas is pretty autonomic in its hormonal regulation. But don't worry, put an X to it. I'll make a note so you can do that right now. So this is a question that we can briefly talk about. I want to try and make it to the end where we have a fun exercise instead of going through me just talking. Why do we have a pituitary hormone, a hypothalamic hormone, and then also finally the hormone that's going to take action, the thyroid hormone? Why don't we all just organize it such as the antidiuretic hormone, the oxytocin? Why not ask the brain to organize everything? Does anybody want to throw out something why that might be? Yes? Signal amplification, that could be a good way. Yeah. It could also be something else, right? If you have more steps that are required to do something, what does that allow you to do at each step? Yes? To regulate, exactly. So that's why I think this happens. If the hypothalamus thinks, oh, this hormone is needed, it secretes it, and then that's it. There's no other way to regulate it. Whereas if it's the right season to breed, but you're really starved of food, those signals are integrated at multiple levels, and you have multiple levels of control to still not go ahead and try and have babies when it really makes no sense. Excellent. So here we have an example of this multi-step control of hormone release. So the end hormone, thyroid hormone, has a bunch of different effects, right? It increases your metabolic rate, it changes the expression of different receptors for other hormones. In tadpoles, it's necessary to get rid of the tail and become a frog. It does a bunch of different things. Very important hormone. Things that might regulate it, stress. In babies, if they're really cold, that would tell the hypothalamus, hey, it's good that we would have some more thyroid hormone. So the hypothalamus makes its thyrotropin releasing hormone, or TRH, sends it on to the pituitary, which has a positive effect on the pituitary. Pituitary makes thyroid stimulating hormone. That goes to the thyroid gland and stimulates it. That's that little plus sign there. And then the thyroid gland finally releases thyroid hormone. This goes into the body and turns on and off all sorts of things, right? This is the effect that we're actually after. Now, if this signaling pathway were to get on and get out of control, that would also not be good. We're spending too much energy. And so there is a way for the system to let higher up in the chain organs know that enough hormone has been made. And we use this term negative feedback. I think we talked about this in earlier lectures too. So if thyroid hormone is made, it can actually bind to the anterior pituitary and negatively affect it. So it'll tell the pituitary, look, we already have thyroid hormone, you can now stop releasing your TSH, right? So then this decreases, this stimulatory effect decreases, and the levels of thyroid hormone don't get out of control. So this is a very classic way that this HP something, HPT in this case, hypothalamus, pituitary, thyroid gland. We have hypothalamus, pituitary, adrenal gland axes, HPA. They all organize kind of in this way, and then there's negative feedback. Sometimes just the pituitary, sometimes also the hypothalamus, just to make sure that we have the right balance of hormones. A thyroid hormone does a lot of things, mostly in us metabolic rate. But if you lack thyroid hormone from early on, your brain doesn't develop properly. You have really severe problems. So it's really an important hormone. It can changes the effectivity of other hormones by increasing the receptors for those. So it can have an accelerating effect. That also means that many things can go wrong. So here's an example of Graves' disease, where there's this immunoglobulin, an antibody that your body makes that binds to the thyroid gland, and it has the same effect as TSH. So it'll tell the thyroid gland that there is TSH, right? That's what it's mimicking. And the thyroid gland responds by secreting thyroid hormone. This TSH normally tells the thyroid gland to secrete thyroid hormone, and it also tells it to grow. It's a trophic hormone, right? And this is what's important in the exercise that we'll do, hopefully, in a few minutes. So this antibody that your body makes isn't what you need, but this is what it does. It mimics the action of TSH. So it's going to go to the thyroid gland. It's going to make a lot of thyroid hormone. The thyroid hormone then goes back upstairs to the pituitary and inhibits. So the pituitary reduces the release of TSH because it is noticing that we have a lot of thyroid hormone. But the thyroid hormone levels don't dry up because this antibody continues to stimulate. It continues to stimulate the thyroid gland, so the thyroid gland would grow, and it keeps secreting thyroid hormone. This antibody, this is an autoimmune attack, and one of the things that is very characteristic is that the eyes really start to bulge out. The muscles of the eye are inflamed, and they get swollen. The osmotic value of the liquid in the eye changes, it increases, so it keeps water in, and that makes the eyes bulge. This is very typical for people that have Graves' disease. So you don't see this with all thyroid diseases. Here's somebody who has a goiter. We saw this in the beginning. And I think this picture was from somebody who lacked iodine in the diet. So there's no iodine in the diet. That means that there's very little thyroid hormone made because that's built using iodine. So the negative feedback is lacking. The system goes into overdrive. Thyroid gland gets a lot of stimulation, so it grows and still tries to make thyroid hormone, but it's still not working. So the system keeps looping back, and the thyroid gland just grows, and becomes really giant. So, 4 minutes. Wow. Here are some cases where we have low levels of thyroid hormone, so hypo, low levels, or hyper, very high levels of thyroid hormone. And they can, yes or no, result in a goiter, this growth of the thyroid gland. And so I thought we should go through one by one and explain to ourselves why we would or would not get this growth of the thyroid gland. Right, so what is needed for the thyroid gland to grow? What signal do we need to see increase? TSH, right? So this is the question that we're asking ourselves. In the case of hypothyroidism, so too little thyroid hormone, that's caused because the thyroid gland is not working, would we see an increase in TSH? Shall we do a raise of hands or shall I open the clickers? I can put the clickers on. So let's put let's put yes A and no B.
Wait for what?
So 9.
Maybe.
Do we get thyroid gland? I think she has. Or no? Do we get TSH?
So I'll move it over to this side. Is that easier? Yes. Yes. So if we have the thyroid gland itself, the thing that makes the thyroid hormone, if that's failing, that's broken, it doesn't make thyroid hormone no matter what we tell it, will we get a goiter? So most people say A, yes, and that's true. This guy is not making thyroid hormone, the negative feedback is missing, so this part goes into overdrive. The anterior pituitary is making a lot of thyroid stimulating hormone. And so that, there we are, yes. Now we'll see a growth of the thyroid gland. So what happens if the lack of thyroid hormone is due to hypothalamus or anterior pituitary not working? Would we see a growth of the thyroid gland? Would we see an increase in thyroid stimulating hormone? I think most people are switching over to no, and that is good. Because either the hypothalamus isn't sending a positive signal to the pituitary, or the pituitary is broken and isn't sending its signal that causes the growth to the thyroid gland. So no. What if we don't have iodine in our diet and we can't make thyroid hormone? Okay, people are changing that. Yep. Now Hashimoto's, an autoimmune disease, is attacking the thyroid gland. Excellent. You guys are doing a great job. Right? The autoimmune disease might be making the thyroid not functional, less thyroid hormone, but it's an autoimmune disease. It's attacking this tissue, so it's going to destroy it and break it down. I'll post the slide with all the answers. I can also quickly open them up right here.